Hello everyone. Welcome to mul-T-omics, an integrated omics approach to T-cell activation. Thank you for joining us. In today's session, we will give you a glimpse of Elucidata and poly-enabled services. We would talk in depth about our latest paper on bioarchive, integrated metabolomics and transcriptomic profiling, reveals novel activation-induced metabolomic networks in human T cells. Before we begin today's session, we would like to show you a 1-minute video on poly-intermics, an integrated omics tool that was useful in the analysis for the paper that is going to be discussed today.
A deeper knowledge of disease is crucial to find targets that succeed in the clinic. And the journey from target to therapy is a long one, as you find yourselves drowning in omics data. But with Intermix, you can sharpen your search with just a few clicks. Intermix uses omics data to get a quick, insightful look at metabolism on a global scale. Generates practical insights better and faster. Intermix uses embedded machine learning algorithms to suggest relevant publications, phenotypic data, and transcriptional data sets. And just in case you need help, our experts are ready to work alongside you. All of this on a user interface invented just for target discovery. So, start stronger, move faster, and dig deeper, and get better actionable results. Intermix. Put omics in your mix.
Before moving ahead, I would like to give you a brief introduction of a brief outline of the session. This webinar is being recorded, and a copy of this webinar will be shared with you over the email. We will take all the questions in the last 10 minutes of the webinar. You can post any questions you want us to answer in the question and answer session towards the end. We also have a support team over here. If you have any doubts or questions during the demo, you can post them on the chat and they will reply promptly. We will start with an introduction to Elucidata by Dr. Abhishek Jha, followed by Dr. Josefa Bandukwala's talk around our publication.
Hi. Hey, good morning, everyone. I hope
Dr. Josefa Bandukwala is Vice President, Platform Innovation and Biology at Siglion Therapeutics. Dr. Bandukwala has over 12 years of experience in biological research across industry and academia, including at the University of Chicago and Pfizer. Coming to Dr. Abhishek Jha, he is founder at Elucidata. In addition to his research experience at MIT and the University of Chicago, Dr. Jha has over 5 years of experience at Agios Pharmaceuticals, where he was a key driver of 2 different FDA-approved drug discovery programs. So I'll hand over to Abhishek now. Abhishek, over to you.
good morning, everyone. We wanted to thank you for taking an hour out of your busy schedules to come and join us this morning to learn more about the most recent publication that we have put out as part of the collaboration between 3 organizations: Pfizer, Yale, and Elucidata. Josefa will be going into details of that piece of work in a few minutes from now. But I wanted to get this conversation going by introducing all of you to Elucidata. And I'm particularly proud of this slide, which this picture was taken in our last company retreat. We have a 60 people company. We are based in Cambridge and in Delhi in India. We have recently expanded also to San Francisco and Bangalore. Over the last 3.5 years, we have worked with 25 different biotech and pharma companies. And this indicates the commitment of our company to bring integrated omics approach across every stage of drug discovery. The group of expertise that we have put together under one roof is very diverse. We have software engineers, bioinformatics scientists, and all of us share the mission of enabling integrated omics approach to accelerate drug discovery. And our worldview is very data-centric. So we are a dry lab, and we start getting engaged once the data has been acquired. And the barriers that we kept hearing and experiencing ourselves firsthand as we tried to take different kinds of omics data to enable discovery led us to come to a place where we developed our innovation framework to address these challenges. What we call that EPIC. EPIC is stands for ingestion, processing, interpretation, and collaboration, which indicates the 4 stages of life cycle of any omics data sets. There are a lot of challenges. Just as you ingest data from a computer that acquired data or a publicly available data set that you want to use, processing has its own set of challenges with a lot of really compelling open source tools available, but they're not quite readily useful for scientists in the lab most of the times. And then, those open source tools have to be customized for your specific needs. Interpretation, that's the job of the expert. Like, that's why we did this experiment in the first place, is heavily manual, driven by a few stakeholders. And we wanted to enable faster interpretation of the data that has been generated. And finally, there are multiple stakeholders of any omics data set. And how do you collaborate between them to get to an insight faster? And together, these 4 pieces provide an innovation framework for what we wanted to do as a company. But we also realized that these 4 challenges are very similar across all omics. So if you're using metabolomics data or proteomic or epigenetic, you'd have the same issues. However, the challenges within each of them are very unique. And that's something that we have learned over the years, and we continue to learn about it, and that gets reflected into what we have built. So the outcome of all our work in the last 3.5 years has resulted in a platform which we call Poly. Poly is a cloud-based platform that enables integrated omics approach in any lab to specifically identify and validate targets. We offer Poly as a licensable workflow where you can license any of these workflows and use it for your own discovery needs, much like in a DIY mode. You can also hire scientists at Elucidata to use Poly on your behalf, which is what we call Poly-enabled service. The presentation that Josefa, the part of the presentation that Josefa will drive, was an outcome of a Poly-enabled service that Elucidata had with Pfizer. Poly, the platform specifically, has many different workflows which address different omics like proteomics, RNA-seq, single-cell RNA-seq, metabolomics, flux data, CRISPR screening, mass spec imaging, and so on and so forth. This list will keep growing as we find more relevant use cases for different kind of data set to drive target identification and validation. On top of these workflows, Poly has a number of very compelling platform features. For example, it allows you to customize these workflows. There are many interesting project management capabilities. Poly ensures that the analysis is reproducible without any additional input from the users. Almost all the apps end up with a pathway level insight, and you have the power to personalize your ML models which underlie these workflows. And finally, collaboration is something that all these workflows on Poly have available to them. Just to get a closer look into the workflows that we have right now, which are available for an annual subscription. They fall in 4 different classes. Bunch of workflows that we have built are around data that comes off mass spec instruments. We have PolyMetscape, which is focusing on untargeted metabolomics studies, PolyPhi for labeling tracer studies, and PolyQuantFit, which helps us quantify hundreds of metabolites in hundreds of samples in a matter of few clicks. Another bucket of workflows that we have use data that comes off sequencing platforms. For example, polyRNA-seq is focused on transcriptional data analysis. PolyCRISPR screening helps take sequencing data, FastQC files, and finds genes that are positively selected or negatively selected in a particular CRISPR screening campaign. We have poly-Int-omics, which integrates 2 different kinds of omics data, in this case, transcriptional and metabolomic, or proteomic and metabolomic. We also are increasingly moving towards bespoke workflows where scientists in lab have developed their own workflows to do, let's say, high throughput screening, data analysis, phosphoproteomics, and so on and so forth. And one of the offerings that we have is where we can take these custom workflows and put that on Poly in a matter of days. And the rationale is that we have built 7 workflows. We'll build 10 more, but there are hundreds out there which are being actively used by scientists in different labs, and we want to enable them to move their workflow to Poly and have access to all the powerful features of the platform. For example, version tracking, project dashboard, collaboration features, so on and so forth. So the way it works is, you prescribe the workflow by specifying the type of data, input files, format, different algorithms or processing that you want to do, dashboards. And 2 weeks later, you would have that on Poly with all the features of the platform accessible to you on day 0. We have applied that approach to bring different kind of workflows onto Poly. As I mentioned earlier, we also offer Poly as an service where you can hire scientists at Elucidata to partner with you to address your research problems. And we have had a pretty good traction around that as well, mostly driven by the fact that we have, as a team, very broad capability in handling different kind of omics data sets. We have also tinkered and played and dug deep into many publicly available data sets that are routinely and increasingly becoming part of early R&D efforts. And finally, we are a multidisciplinary team who work very closely with you to enable insight generation. And a lot of the themes that I'm touching upon today are reflected in the case study that we'll just demonstrate. So many of the workflows that I mentioned on Poly were used in this work that is now out as a preprint on bioRxiv. And I hope that that case study illustrates the power of collaboration with us. And beyond this single case study that we'll look into more in depth, we have partnered with different, more than 15 industrial and academic labs over the years. And that has resulted, among many other things, some publications and posters in different conferences. All of this has happened because of a team of around 20 data scientists and bioinformatics scientists, supported by a very strong team of engineers who have been developing Poly and continue to do that. We're very excited to have a very passionate team, people who are deeply engaged, very rigorous scientists, who work with you to drive your discovery efforts in close collaboration. Now, I hope this sets the stage of what we are as a company, and I'll now hand it over to my colleague and friend Josefa, who was the leader of this collaboration when he was at Pfizer. So again, just to be very clear, the work discussed in today's presentation was a 3-way collaboration between Pfizer, Yale, and Elucidata. Josefa, when he was at Pfizer, worked with us, and that part of that work has now been available as a preprint on bioRxiv. So with this setup, I'll hand it over to Josefa.
Hello. Hello, can you guys hear me? Okay, great. So I'm going to take you guys through the publication that we put out on bioRxiv recently, just the way Abhishek described. So thank you very much, everyone, for coming and listening to us. Before I go on, I just wanted to take a moment to really give acknowledgement to the Pfizer postdoctoral program and its commitment to cutting-edge research in science. The entire efforts that I'm going to describe to you were done as part of the Pfizer's postdoctoral program and funded by that. And they are the work of Samantha Hema, who was the postdoctoral research fellow at the Inflammation and Immunology Research Unit. And her efforts were really in close partnership with Shashank Jadhav, who was the senior data scientist at Elucidata. And I think the phenomenal work done by both of these has resulted in the publication, which is now on bioRxiv. And I'm really excited to tell you a little bit about the thought process behind what motivated us to interrogate transcriptional networks in human T cells and what some of our key learnings were. So as you probably are aware now, that immunometabolism is a field that has taken the entire community by storm. There's been a lot of excitement around this field. And some of its excitement stems from some pioneering the work that has been done in academia for a very, very long time. And essentially, the realization has come that the metabolism of a cell really dictates its function. And specifically immune cells, when they are either in the resting state or they get activated, they can switch the entire metabolic profile. And this switching of metabolic profile is really required to determine the fate that an immune cell will take. As you are probably aware, most immune cells can take either a tolerogenic or an inflammatory state. And the key learnings that were coming was that this fate switch in function reflects also a fate switch in their metabolism. So at a very, very broad level, generally cells can take on one of two metabolic states: an anabolic metabolic state, where they are really increasing their biomass and proliferating and actively producing biomolecules. And then the other state that they can take is one of catabolic metabolism, where essentially this is featured with a hallmark of mitochondrial respiration. There's fairly little glycolysis going on, and almost all of the ATP that is generated through oxidative phosphorylation is generally used for housekeeping and maintenance processes rather than biosynthesis and growth of the cells. The key findings that were coming out is essentially this anabolic state of immune cells. And essentially it was a unifying feature, not only just restricted to lymphocytes, but even things like macrophages, and more important, even non-immune cells, such as synovial fibroblasts that participate in the inflammatory diseases, were found to switch their metabolic state towards anabolic metabolism to drive inflammatory functions. And so the very natural question that happened was, what is the genesis of this metabolic switching? And is this actually part of normal immune homeostasis? And so there was increasing data that was turning around to actually suggest that this was the case. And here is a diagrammatic representation of that, where multiple processes that we are all aware of that are really, really critical for us to maintain immune homeostasis were being mediated through metabolic regulation of immune cells. Let me give you a few examples. So one of the classical places where immune homeostasis is kept in very, very close control is during pregnancy, where the fetus is foreign to the mother, but the mother doesn't mount an immune attack on the fetus and is actually immunologically silent. And one of the key mechanisms by which this happens is that the maternal trophoblast, which is pictured in the box to the left, in the lower part, the maternal trophoblasts secrete an enzyme called IDO, indolamine deoxygenase, which degrades tryptophan, which is a key amino acid that is required for T cells to mediate the inflammatory functions. Regulatory T cells are another means of mediating immune homeostasis. They actively block the inflammatory functions of effector T cells, and more studies had shown that one of the major mechanisms by which they do so is by expressing ectonucleodases like CD39 and CD73 that actually deplete ATP in the T cell milieu. And of course, the most famous example is that of myeloid-derived suppressor cells producing enzymes like arginase, which again deplete the nutrient environment around the T cells. So essentially, the consensus that was building in was that this was the natural mechanisms by which immune homeostasis was being maintained in healthy individuals. And so the question that we had from the translational perspective was that could we harness some of these power processes to actually induce therapeutic immunomodulation for people that was for patients that were suffering from inflammatory and immune-mediated diseases. And so the question was that if we could alter the metabolic state that was maintained in a highly anabolic state in inflammatory diseases back to that of a resting, quiescent, catabolic state, would we be actually able to either A, completely turn off the inflammatory T cell responses, or B, amplify the natural immunoregulatory mechanisms that are mediated by Tregs, or C, finally, which is use them as a combination therapy so that we could even further potentiate the effects of currently marketed immunosuppressive drugs. And so as a proof of concept, there were some papers that had already published, essentially from Lawrence Morell's lab, where they had shown that in a mouse model of lupus, which is the triple congenic mouse model, this is a naturally occurring mouse model of lupus where anti-nuclear antibodies develop. There is splenomegaly, which happens over a long period of time. And what Lawrence Morell's group demonstrated was that treatment with two metabolic regulatory drugs, metformin, that is heavily used in type 2 diabetes, which reduces systemic glucose levels, and 2-deoxyglucose, which is actually just simply a glucose analog and blocks hexokinase, the first step in glycolysis, could result in dramatic inhibition of the inflammation that is associated with lupus, with the mouse model of lupus. And so I think the most telling data in that slide was the splenomegaly slide, which is pictured in the lower right. And what you can see is that in the control triple congenic mice, you have massive splenomegaly, the spleen becomes huge, but simply treatment in the very next spleen that is pictured over there, treatment with these two metabolic drugs, metformin and 2-DG, was sufficient to reverse the splenomegaly. And this was actually backed up by some clinical observations as well, where when patients were looked at, lupus patients were analyzed, what they found was that T cells isolated from lupus patients actually had a higher glycolytic rate, which is depicted by their extracellular acidification rate over here. So essentially, there was evidence of both that in the clinic, inflammation was accompanied by anabolic metabolism, upregulation of anabolic metabolism in immune cells, and that if you were to intervene and block that, you could actually reverse diseases in a preclinical model. So the real question that we had was that, well, this is all really well and good, but there is one little missing component, is that we have clinical data that is observationary, and the perturbational data that we have is in mice. But would you have a similar effect of perturbation if you were to block the inflammatory the anabolic metabolism of human T cells? And so essentially for those experiments, we turned to the NSG mouse model, in which you can implant you can have a functional human immune system in these mice that are deficient in their own immune system. And so in this experiment, what we had done was that we transferred human PBMCs into these NSG mice, which is pictured in the schematic representation on top. And after 2 weeks, we just followed the mice, and the mice were either left untreated or were treated with the same cocktail of metformin and 2-deoxyglucose. And very similarly to the data that were demonstrated from Penny Morell's group, what we found was that treatment with this combination of metformin and 2-deoxyglucose was also sufficient to block inflammation that was associated with the human immune cells recognizing mouse antigens as foreign and mounting an immune response towards the skin, the kidneys, the liver. Over here, I'm just showing you a histopathological data of the skin. And here is healthy looking skin, which is now being shown to you in the top right, first box. If you put in immune cells, human immune cells in these NSG mice, they get heavily inflamed, which is in the middle box. But then if you treat with metformin and 2-deoxyglucose, you can completely reverse that phenotype. And then in the bottom graphs, what you can see is that this is accompanied by a decrease in human interferon gamma production from the T cells, which is a key inflammatory cytokine that is being produced in these models. So essentially, if I were to summarize some of the data that I have shown you so far, we were able to demonstrate that the metabolic state of immune cells really drives their inflammatory functions. And then if you pharmacologically suppress this anabolic metabolism through use of molecules such as metformin and 2-deoxyglucose, you can actually attenuate the pathogenic inflammation driven by both murine and human immune cells. So a key question that was remaining right now for us was that, well, metformin and 2-deoxyglucose are pretty broad metabolic regulators, but what was the exact metabolic pathways that were being turned on in immune cells, and specifically in T cells upon activation, that were requisite for their inflammatory functions? And so to look at that, we followed on the footsteps of a lot of academic work that had been done in murine T cells, but was a little bit lacking for human T cells. So we decided to start at a very, very fundamental level, and using the CHOS instrument, we basically asked the question, does the metabolism, the switch in metabolism in human T cells reflect the same that has been observed in numerous murine studies? And so what you can see over here is the extracellular acidification rate, which is a proxy measure for the amount of glycolysis that the cell is undergoing, aerobic glycolysis, in which glucose is taken up, but a substantial amount of that is then secreted as lactate, which leads to the acidification of the media. And we basically measured the glycolytic rate in cells that were in human T cells that were isolated from peripheral blood of healthy donors in either the resting state, which are the open circles, or then activated by in vitro activation with anti-CD3 and anti-CD28 stimulating antibodies, which is now depicted in the closed black circles. What we saw was that, as had been shown previously in murine studies, even human T cells upon activation demonstrated a massive upregulation of glycolysis. But what was really surprising to us was also that this upregulation was not limited to glycolysis, but also mitochondrial respiration was heavily upregulated, which is now being represented in the graph in the right. So OCR is the oxygen consumption rate that is being monitored. And again, you can see that activated T cells basically have a very high oxygen consumption rate, which means the mitochondrial respiration is moving at a very rapid state. But what came as a surprise to us is that if you added glucose to the media, rather than further amplifying the metabolic about the mitochondrial respiration, it led to a decrease in mitochondrial respiration. And when we actually took a step back and looked at the data, what we found was that both of these phenomena had been described in literature, specifically in cancer cells, and the exaggerated glycolytic metabolism was obviously known as the Warburg metabolism, and the suppression of mitochondrial respiration upon addition of glucose was the classical Crabtree effect that had been described. And so essentially what we found was that activated human T cells upon activation exhibit the hallmarks classical hallmarks of cancer cell metabolism. What was even more intriguing to us was that activated human T cells, while they consume tremendous amounts of glucose, they are not using that to fuel mitochondrial ATP production and mitochondrial respiration. So a key question that was left in our minds is that, are there other substrates that the T cell utilizes? And what is the role of glucose? Why is the activated human T cell consuming such large quantities of glucose? And so to address those questions, we turned to a classic labeling experiment where we actually took these T cells, kept them in either resting or activated state, but then we added isotopically labeled glucose, uniformly labeled glucose, or glutamine to these cultures. And we followed the glucose label through the various metabolic pathways, both through glycolysis and central carbon metabolism. And now we are focusing towards the panel A, which is on the left. And what we found was that activated T cells massively upregulated their consumption of glucose. That was well reflected from our seahorse experiments as well. But what was surprising was that the proportion of glucose label that was flowing through the various steps of glycolysis was very, very similar. So if we were to look at the circles that represent the total pool of the metabolite pyruvate, the size of the circle is much bigger, suggesting the total levels of pyruvate are much more in activated T cells. And the pie chart in the middle shows the percentage of label that is coming from the glucose that we had fed in. And you can see that it's a very, very similar proportion. So essentially what that meant is that the cells were augmenting the ability to take up glucose, but they were not reprogramming the way in which they were utilizing the glucose as it went through the metabolic pathways. The second thing was that as we stepped down to central carbon metabolism, what we found was that through the successive steps of the TCA cycle, the glucose label was diluted. And this could mean one of two things, that essentially the glucose label was being transported out of the citrate cycle, or that there were other carbon sources that were feeding into the TCA cycle, diluting out the glucose label. So with that thought process in mind, we turn towards looking at glutamine, which is another carbon substrate that can very rapidly be used to feed the TCA cycle through conversion through glutamate. And what we found was very similar to glucose. Upon activation, there was a huge upregulation in glutamine consumption by the activated T cells as well. But in contrast, what we found was that there was a massive reprogramming in activated T cells on how the cells utilize the glucose. So as you can now follow the pie chart, you can see that the proportion of glutamine label in the various metabolites, glutamate and succinate, is very different in the activated versus resting T cells. So overall, these data told us two things. Is that there are various carbon sources that can feed into the TCA cycle, but there is a substantial reprogramming upon activation of T cells of how they handle the glutamine carbon sources as compared to that of the glucose carbon sources. So to get a better handle on that, we went back and did a transcriptomic profiling to look at all the genes that were changing upon T cell activation, all the metabolism associated genes that were changing upon T cell activation. And essentially what we found was that they were clustered along the kinetics in which these transcriptional changes happened. And there were some genes that actually were downregulated, but on the whole, the majority of metabolic associated genes were upregulated either in a prolonged manner or in a transient manner. So the comparisons of the profiles represented by T2 and T3 respectively. And this changes in the transcriptional level of genes was also accompanied by a very similar changes in the various metabolite levels that were there inside the cell. But what was really lacking at this point was we had no idea of how these two data sets talked to each other. So essentially, if I were to summarize what I have told you so far, what we found was that there are a huge number of similarities between mouse and human T cell metabolism. Upon activation, both glycolytic as well as mitochondrial metabolism is upregulated in human T cells. The TCR stimulation leads to utilization of not just glucose, but other carbon sources such as glutamine as well. And these seem to have different fates inside the T cell. And that the modulation of T cell metabolism, the huge shift that we see in regulation in metabolic reprogramming in T cells, is not only at the level of substrate consumption, but is also accompanied by active transcriptional regulation of multiple metabolic enzymes. And the key question, as I had mentioned on the previous slide that we were left with, was trying to understand of how do we integrate both the metabolic data sets as well as the transcriptional data sets that we have so that we could get real insights into some key nodes that are required for metabolic reprogramming of T cells and also for regulating the effector functions of T cells. This is where we found Elucidata to be a phenomenal thought partner for us. We had all of these huge data sets, but trying to integrate them together in silico was a little bit of a challenge. And Dr. Jha had recently published a paper where they had successfully integrated both metabolic and transcriptional data sets. And they had found that this actually revealed metabolic modules that were required for mouse murine macrophage polarization. And so we asked the question if we could use a similar approach to understand the metabolic reprogramming that was happening in human T cells. And so here is the data set. And essentially, I'm going to take a minute. Whoops. Just go back for a second. So this is essentially the same data sets that I showed you before, the transcriptomic and the metabolomic data sets, but integrated through an approach which they have coined as CombiT, which is the concordant metabolic and transcriptomic integration. And what we actually found when we looked at this data set was that the picture was quite different than what we had canonically thought of based on just regular textbooks. This was not a classic textbook view of metabolism that we were looking at. Before I go into the depth of this data, I just wanted to give you a little bit of a key on how this data is being represented. So what we are doing over here is we are comparing the metabolism pathways in resting and activated T cells. So essentially what this is a fold change. It's a difference in pathways, both in metabolite expression and transcript expression in resting and T cells that have been activated for 24 hours. The color, red versus blue, talks about whether a gene is being upregulated or downregulated. The thickness of the line basically talks about the magnitude of the significance of how significant that change is across multiple different donors. And essentially in this plot, red denotes genes and metabolites that were upregulated upon activation, whereas blue is those that are either high in resting T cells or for that matter down-regulated upon T cell activation. So as we look through this plot, I wanted to kind of anchor you a little bit by looking at glycolysis. So this is the train that we had been following, the train of thought that we had been following so far. Glycolysis is heavily upregulated upon T cell stimulation. And glycolysis begins with, as we think about it, glucose, which is being depicted in the graph in the upper right corner. And then as we follow the glucose down, it goes through classical glycolysis, getting converted to glucose 6-phosphate, all the way down to GAPDH, enolase, and pyruvate. And you can see that this entire pathway is highlighted in red, suggesting that this pathway is being upregulated upon T cell activation. But right then and there, essentially, we found there to be some really interesting thoughts. And so the way this analysis works is rather than overlaying the data on a canonical view of central carbon metabolism and glycolysis that is depicted as the line and circle model in most textbooks, what this representation does is tries to find connections between pathways and nodes that are being most dramatically modulated upon activation. And what we found was that as we looked through that, it was not the linear stick and ball model, but essentially that there were multiple inputs and multiple outputs going out from these pathways. So, for example, it turned out and looked from here that glucose was not only the only hexose that could go through glycolysis, but there were other hexoses as well that could be converted into glucose and used for glycolysis in activated T cells. And the pathways for those conversions were also being upregulated. And the implication of this data was that modulating glucose metabolism may not be sufficient because other hexoses might be able to compensate for it. Also, modulating the early glycolysis may not be sufficient to regulate the effector functions of T cells. On the other hand, what we also found was that as we looked at the TCA cycle, again, it did not come up as a classical circle, but it seemed to have many branch points associated with that. And what we saw was that there were multiple pathways that could either feed into the TCA cycle or it could actually be being fed out of the TCA cycle for biosynthesis. And so essentially what we felt was that there were a lot of amino acid pathways that were heavily being modulated upon T cell activation. So it was not just glutamine, but glutamate metabolism, arginine metabolism, proline metabolism, all of these seem to be heavily regulated upon T cell activation. And further experiments would be required to figure out actually the directionality of these processes. So are proline, aspartate, arginine actually being utilized as carbon substrates to fuel TCA cycle intermediates? Or is it that the TCA cycle mediates are used as precursors to generate biosynthesis of these amino acids? But what was really, really heartening to see in this entire analysis was that targets of canonical drugs like methotrexate, which is the dihydrofolate receptor, DHFR, which is pictured over here, as well as purine metabolism, which is a target of a lot of immunosuppressive drugs such as azathioprine, leflunomide, were all demonstrated to be modulated upon T cell activation. So the question that we had on our minds at this point was that while this is a very powerful view that we have upon the changes in metabolism that are being accompanied by TCR stimulation, the TCR stimulation that we are giving to the cells is highly artificial. So inside the body, essentially, it's an antigen-presenting cell which presents a peptide in the context of its MHC to the TCR, and that leads to the activation of T cells. In contrast, what we had been doing was stimulating the T cells with anti-CD3 and anti-CD28 antibodies, which have a dramatically different affinity for the TCR than the peptide MHC complex. And so the questions on our mind was essentially, does this mode of activation really impact the effector functions and in turn the metabolism of the T cells? And so to look at that, while we were not set up to actually look at the metabolic changes in T cells in a co-culture model, what we decided to do was to use different formats and modes of TCR signaling that we knew had a different impact on effector cell function, and then in turn look at the metabolism that went along with the T cells. And so over here, we essentially use two reagents. One of them is called immunocult. Again, this is a proprietary formulation by Stem Cell, which uses anti-CD3 and anti-CD28 stimulating antibodies, but rather than having them immobilized on a hard plastic surface, these guys are cross-linked by a proprietary polymer. So that is the data that is shown in the green. And then in the blue, we use the classical plate-bound method of co-stimulation, which we know leads to a very robust and strong activation of T cells. And what we actually found over here is that these two different modes of TCR signaling had a pretty dramatic effect on the effector functions of T cells. Whereas strong TCR stimulation through plate-bound format led to an upregulation of cytokine production, which is pictured in the panel C, with looking at interferon gamma, IL-2, and TNF. And you can see that in all cases, the blue lines are higher than the green lines. And I'd just like to point your attention that it's a log scale that we are looking at. So in all cases, plate-bound stimulation led to a more significant upregulation of cytokine production than immunoccult stimulation. On the other hand, when we followed the proliferation of the T cells, what we found was that the immunoccult-mediated stimulation gave slightly better proliferative signals to the T cells than the plate-bound stimulation. So we definitely knew that we had two formats of T cell activation that differed in the effector functions that were mediated by the T cells. The question that we had on our mind at this point was, well, does that translate to different metabolic networks and nodes inside the T cells? And so we went back and looked at that through our Combi-T approach lens. And so here we are looking at pathways in greens are the ones that we saw come up specifically when the cells were stimulated with immunoccult, and the lines in blue are the ones that specifically came up when the cells were stimulated with plate-bound activation. And what we found overall was that overall very similar pathways, as I mentioned to you before, amino acid metabolism, glycolysis, proline metabolism, 1-carbon metabolism were being observed irrespective of which format was used to stimulate the T cells. However, we did see some really intriguing differences, is that depending on the mode of such stimulation, different routes or metabolic networks were being engaged to catalyze similar biochemical transformation. What do I mean by that? So if you look towards the area which talks about 1-carbon metabolism, where glutamate is getting converted into formate, you can see that when you use the immunoccult activation, essentially the canonical pathway that is mediated by DHFR seems to be very, very strongly engaged. However, with immunoccult, it seems to utilize a lot less biochemical enzymes and is mediated by these enzymes GGT1 and ESD. So there were differences that accompanied plate-bound versus immunoccult activation, both not only in terms of effector functions, but also which metabolic networks were being engaged in the T cells. So if I had to just take a moment and summarize the data that I have shown you, what we found is that looking at this combinatorial integration of distinct data sets, transcriptomics and metabolomics, has the potential to reveal key nodes that are changed in T cells upon activation. And essentially, distinct formats of TCR signaling can lead to different metabolic networks. And so the implication of all of this data is that there is an urgent need to actually study the physiological context of T cell activation, which is in vivo, an antigen being presented in the right context with a professional APC to T cells, to understand the key metabolic pathways that underlie the pathological functions of immune reactions. So finally, what we wanted to do was kind of understand a little bit better that if we were to perturb these signaling cascades that were being induced by TCR stimulation, would we be able to change the metabolism of the T cells? And so we looked at two canonical pathways, mTOR activation, which we know is a central regulator of metabolism in a lot of different cells, as well as ZAP70, which is a key kinase that is required for TCR signaling and is really, really proximal to the T cell receptor signaling. And in the interest of time, I won't go through all of the data, but essentially what we found was that it seemed that TCR stimulation utilizes multiple signaling cascades to regulate the huge plethora of metabolic enzymes that are being regulated upon T cell activation. So essentially, whereas mTOR inhibition was only able to deplete a subset of metabolic changes, ZAP70, basically blocking TCR stimulation in general, had a much more dramatic effect on the T cell metabolism. And this was borne out not only in our functional genomic approaches, but also upon pharmacological modulation. So over here, we are looking at the changes in transcriptometabolomic networks in T cells when they were treated with either rapamycin which is an inhibitor of mTOR activation, or cyclosporin, which blocks the NF-κ family of transcription factors. And what we found was that utilizing these two different modulators had a very distinct impact on the transcriptometabolomic networks, with NFAT having a much more dramatic effect. And so we have always thought of NFAT as a transcription factor that predominantly regulates cytokine genes. But what our data reveals is that in addition to cytokine genes, it also regulates a number of metabolic enzymes inside T cells. And finally, the last data slide that I'm going to talk about is that we did an experiment where we combined both rapamycin and cyclosporin to see if combinatorial treatment, which we know has a very, very strong impact on the effector functions of T cells, would have a greater impact on the metabolic networks in T cells. And once again, we found that the metabolism of the T cell and the effector functions were very coordinately regulated, where we had a most dramatic impact of the combination of those drugs rather than either drug alone. So finally, what we conclude from all of the work that we have done is that the CombiT approach is a very powerful platform to query metabolic changes that are associated with immune cell activation and function, and that the efficacy of a lot of our clinically used drugs, in part, could be due to their effects on the metabolic pathways inside T cells. And then from a further development perspective, combination therapies that are well designed so that they can block cytokine production as well as key metabolic enzymes could provide a greater therapeutic efficacy, specifically in disease and intractable diseases like lupus, which require some urgent innovation for patients. So with that, I'll conclude my presentation and be happy to answer any questions.
We have 8 more minutes before we would wrap this up, and we have had some questions along the way. So what I'll do now is I'll read off the question and the name of the person who has had this question, and assign either Josefa or Shashank or myself to respond to them. So the first question is from Bharat, and the question reads, what imparts specificity of generic drugs like metformin and 2DG to T cells? Based on what is known, even other cells, immune and non-immune, should be affected. Josefa, would you want to respond to this?
Yeah, Bharat, that's a great question. And I think the answer is that we don't have any data right now that suggests that it is a specific effect to T cells. You very rightly point out that both of these drugs could have an impact on multiple cell types, not only T cells, but also liver cells. And we know that there is a close nexus between metabolism, systemic metabolism that is mediated by the liver, and immune cell functions. So what we could be seeing is very, very likely a combination of both of these processes. As we move forward and look into therapeutic space, I think the question is on point. We'd have to try and figure out pathways that are very specific to immune cells so we could have a more surgical intervention for the metabolism of these cell types.
Great, but yeah, I'll just reiterate what Josefa said. We have no evidence right now, and we are not making the point that treatment of these are specific to T cells. Moving on to the next question from Tushar. I read out his question. Once the metabolic switching has happened in T cells upon activation, would it convert to epigenetic modifications? And if so, is this reversible, or are we preventing non-activated T cells from switching? Josefa, I'll ask you to respond to this as well.
Sure. So Tushar, great question. So I think that there is a very close nexus between metabolism and epigenetic modulation. So we know that epigenetic modulation requires methylation and acetylation. Both of these require substrates like SAM, which come from metabolic pathways. And so going back to your question, I think that prolonged metabolic switches will result finally in epigenetic modifications as well. And I think that it is a circular process where epigenetic modifications of key metabolic enzymes could also fix the metabolic state of the T cell. And so the second part of your question is, are we preventing non-activated T cells from switching? And I think the answer to that would be, it would depend on the the perturbation that we are using. So for example, if we go back to our data, the mTOR inhibitors seem to work only in activated T cells. They have very little effect in resting T cells, which makes sense because the mTOR pathway is actually not being amplified in resting T cells. On the other hand, NF-κ inhibitors seem to have a much broader impact both in resting and activated T cells. So it is quite likely that depending on the drug that we are using, we could have an impact that is either restricted to activated T cells or more broader to both.
So in yet another biological system, very different from what we just discussed today, when upon IDH mutation, there's a 2HG abundance which leads to epigenetic modifications. And upon, if you bring down the 2HG levels, the epigenetic modifications do get reversed. So there's evidence in this case and many others where there's an interplay between metabolomic rewiring and epigenetic changes as well. Again, a great question. I'll move on to the next one from Zen. For your study using human T cells, do they come from fresh blood or frozen blood or human tissue or purchased T cells? I can respond to this, and Josefa, feel free to chime in.
Yep.
This entire study was done on human-derived PBMC cells from an internal Pfizer program where they sought healthy donors to contribute the samples. So, Josefa, do you want to add something to that?
No, I completely agree. Just wanted to put in a quick point that we have actually done some studies that we did not put forth in this presentation, which were done with both fresh, frozen blood, and purchased T cells, everything that you mentioned. And what we find is that these changes in metabolic reprogramming that we talked about are uniformly seen no matter what is the source of the T cells. So it seems to be a very conserved phenomena that overrides all of these in vitro conditions.
To the next question from Claudia. I am also interested in the cost of Elucidata platform, much like Zenzen is. So Claudia and Zenzen, I can respond to that. We will be in touch with you shortly after this webinar about any such interest. And again, we appreciate your question and interest in Elucidata's platform. Next question from Bharat. I'm repeating. Would autoreactive T cells compared to non-self-reactive T cells have differential bias towards cytokine production versus proliferation based on peptide MHC affinity? Josefa, I'll let you respond to this.
Yeah. So I think it's a great question. I don't think we have the data to really dissect that out completely in human T cells. But from mouse models, what we have found is, yes, that there is definitely a bias towards the impact of peptide MHC affinity and effector T cell function. All of this data comes essentially from work that has been done with altered peptide ligands. What we do not know yet is that in the later stages of inflammatory diseases, how much actual proliferation happens. So what I mean is once those autoreactive T cells have entered inside the target tissue that they are destroying, inside pancreas, for example, in type 1 diabetes, whether the only thing that is important is cytokine production or is proliferation important as well. So I think the context of that would depend upon not only the peptide MHC affinity, but also the differentiation state of the T cells and the localization, the anatomical localization in which they are.
We have less than 2 minutes now, so I'll take one last question, also from Bharat. Has anyone compared ex vivo primary T cells, OTI, or co-cultured with OVA-loaded DCs, either high affinity peptides versus low affinity peptides for T cell proliferation versus cytokine production?
Yes, Bharat, I answered that question a little bit in your, number 6. Yes, it has been done, and a lot of literature on altered peptide ligands. And what we do not know is that it goes exactly along the way that you talk about, where there is a differential impact on proliferation versus cytokine production. What we do not have is anybody actually going back and looking at the metabolism of those T cells as they get stimulated with different altered peptide ligands. So I think it's definitely a very intriguing question.
We'd like to thank everyone for joining us and participating in this webinar. I would also like to thank Josefa for talking about a work that has been a lot of effort from a lot of people, Sammy and Shashank. There are a bunch of other authors without whose contribution we would not be here. Again, very thankful, and also thank you for showing up. We will be sharing the slides and the video recording of this webinar with all the attendees. And for specific questions that we have heard today, we will be in touch with you shortly after.
Thank you so much, everyone. Appreciate your attention.